Hello everyone, I'm Seonju and today I'm going to present the paper. The title is Quorum Catching Bacteria Isolated from Industrial Wastewater Sludge to Control Membrane Biofouling. The context of this presentation, like here. First, I'm going to talk about the background knowledge of this paper and methodology and result of them and the main point of this paper, and finally I will wrap up with the further research. First is introduction. Before start, I want to ask something. Did you know that animals or plants do communicate with each other? And how about microorganisms? Well, they actually do the communicate with each other. I will talk it later. First is about MBR. MBR is a membrane bioreactor. It's a process that combines biological treatment and membrane filtration. There is a many good points with MBR, but there is a huge main drawback called membrane fouling. Membrane fouling is a clogging of pores of membrane, like here, and is related to formation of biofilm, like here. And the formation of biofilm is also related to the quorum sensing. It's a communicating system of microorganisms to coordinate their group behavior. And to limit that, the strategist called Quorum Catching has invited. Next is methodology. First, they use the method of isolation. Isolation is a method to spotting and catching the specific functioning microorganism, like here. They took the samples from wastewater treatment plants in Korea, and they used the chemicals that plays a role in quorum sensing as a language. Like here, I will call it language chemicals. Yeah, so they use language chemical to find the new quorum catching bacteria. Next, also they found the tens of new QQ bacteria, and through the RNA sequencing, RNA analysis, they found the name of them, like here. And next is QQ activity test. After we found the new QQ bacteria, we need to know how well they actually do perform of quorum catching. So they grew QQ bacteria with language chemicals and toxicant called Tama and QQ bacteria. And they measured the QQ activity with the luminescence, the light of reporter strain called A136. They emit the blue light, like here, with the existence of language chemicals. And next is about result of that. This is a picture of isolated QQ bacteria, and among them SDC A8 and SDC U1, they did the best performance of quorum catching. The next graph is about QQ activity test. In graph A, here, the x-axis is about time, and y-axis is the concentration of the language chemicals. As you look at the black line here is a control. The control didn't degrade the language chemicals, but the red line and blue line here, it sharply decreased. It means that they did degraded the language chemicals. And next is the bar graph. The X is many types of chemicals, language chemicals, and Y is the degradation rate. As you look at here, sometimes the blue bar is higher than the orange bar, but generally the orange bar, which means SDCU1, did more fast degraded the language chemicals. And next graph is also about the QQ activity test, but with and without the TAMA, the toxicant. As you look at here, SDCU1, with or without the TAMA, they well degraded the language chemicals. The main point of this paper is first they found the new Chrome-Cancy bacteria from a real ambient plant, ambient sludge, that treated the industrial wastewater containing the TAMA. And second, Bacillus SLC1 and Bacillus SLC-A8 show high QQ activity with or without TAMA. And their QQ characteristics and high growth rates make them promising candidates for mitigated biofouling in ambiers of ambient biofouling.